Hi, I'm Emilie Gauthier, and I'm here to talk to you about the reason you are functioning. Cells. They are often overlooked but are the basic building blocks of all organisms. I will begin by explaining the history of cells and how humans discovered them, and the development of the cell theory, which is basically the constitution of all cells. I will also transition into talking about cell morphology, aka the shape, structure, form, and size of cells. In microbiology, cell morphology is essential to understand to be able to identify certain bacteria. Now let's get started. The discovery of cells. Cells were discovered in the 17th century by a man named Robert Hooke. Hooke discovered cells through a honeycomb-like structure in the slice of a cork. He noticed compartments and called them cells. This piece of cork, he observed, was a dead piece of tissue, so he was only able to see cell walls. Hooke's observation inspired other scientists to study cells, which later on became known as cell biology. It wasn't until years later when living cells were seen by a Dutch biologist named Anton van Leeuwenhoek. He made a special lens in order to observe pond water in hopes to find some evidence of living cells. 169 years later, after multiple scientists continued to view cells and study them, the cell theory was finally accepted in 1839. In 1839, Theodor Schwann concluded that cells make up the tissues of plants and animals. Although plants and animals are made up of cells, the cells were different in structure. He came up with the three basic principles of cell theory. 1. All living organisms are composed of one or more cells. 2. Cells are the basic unit of structure and organization in organisms. 3. Cells arise from pre-existing cells. Schwann was not alone in influencing others to accept the cell theory. He had help from several other scientists named Schleiden, Remak, and Virchow. The cell theory was later modernized and added three more hypotheses. 1. Energy flows within the cells. 2. Genetic information is passed on from one cell to the other. And 3. The chemical composition of all cells is the same. So all this talk about the cell theory, but we haven't even discussed cells themselves. So, what exactly are cells? According to the cell theory, cells are the basic building blocks of all living organisms. Although there are multiple types of cells, they all have two things in common: a nucleus region and a membrane-bound organelles. Cells are the smallest independent component of an organism. They can replicate independently. There are two main types of cells: prokaryotes and eukaryotes. Prokaryotic cells are simpler than eukaryotic cells. They have no nucleus, only a nucleoid. A nucleoid is a region within the prokaryotic cell that contains the genetic material. They are single-celled organisms. Some examples are archaea, bacteria, and cyanobacteria. They contain DNA or RNA, and they are reproduced by binary fission. Now on to eukaryotic cells. Eukaryotic cells contain a true nucleus. They are significantly larger than prokaryotic cells. Eukaryotes contain a plasma membrane which controls the transportation of things in and out of the cell. It is also the way that cells can communicate with each other. This is important for eukaryotic cells because they are mostly found in multicellular organisms. These multicellular organisms are plants, fungi, protozoans, and animals. They are able to reproduce sexually and asexually, or mitosis and meiosis. Although eukaryotes are found in both plants and animals, they are not the same within each. In a plant cell, a eukaryote has a cell wall, contains chloroplasts, and vacuoles. The animal cell does not contain any of these because they are specific to plants only. Microbiology takes a closer look at prokaryotes and certain characteristics that prokaryotes have that are important when studying bacteria. Some of the prokaryotic cells form a layer of carbohydrates or sugars or saccharides, which are identified as a glycocalyx. This layer protects the cells from hosts and infections. It can be different forms depending on how condensed the glycocalyx is to the cell wall. If it is very tight, it can be called a capsule, but if it is very loose, it will be referred to as a slime layer. There are two main functions of the glycocalyx. One, it causes the bacterium within the glycocalyx to become invisible and to host immune systems. This invisibility makes it harder for the phagocytes to surround and engulf it. Infectious strains of bacteria like Staphylococcus, Streptococcus, and Pneumonia all rely really heavily on the glycocalyx compared to the non-infectious strains. The second main function of the glycocalyx is a protective layer that is formed to cover and protect the cell, which makes it hard to kill. This layer is called the biofilm. Some examples of a biofilm are plaque on teeth or pond scum. They both are essentially just buildups of bacterium. Most eukaryotes are present in larger organisms, but sometimes they are microorganisms. The eukaryote microorganisms are fungi, protozoa, and simple algae. Flagella and cilia are two important components to eukaryotic cells. Although prokaryotes have a flagella as well, they have a different purpose in eukaryotic cells. Eukaryotic flagella is more complex than the prokaryotes and functions as a way for the cell to move by causing a whipping movement. Cilia cause a back and forth beating around the cell. It can function as a form of protection, but also assist with the transportation of the cell. Cell morphology is basically just identifying the shape, structure, form, and size of cells while they are being identified under a microscope. It is important to identify the morphology of the cells because it can sometimes indicate that there is a contamination, infection, or health problem. There are three main types of morphology: cocci, bacilli, and spirilla. Cocci is more of a circular shape, while bacilli is more like rods, and spirilla is squiggly. Within each of these categories, there are arrangements. Cocci has single, diplococci, which is 2, streptococci, which is several in a row, tetrad and sarkenes, which is 4 together, and staphylococci, which is a large cluster. Bacilli has single, diplobacilli, which is 2, streptobacilli, multiple in a row, and palisade, which is a large cluster. Spirilla only has single. On the next slide, there's an enlarged picture of the smaller one on this slide. This picture includes the different types of morphologies as well as their types of arrangements. It is from biologydiscussion.com. Each arrangement has a particular meaning behind it. When viewing a slide and you see a diplococci arrangement, it is most likely that it is indicating some kind of bacterial infection. However, this must be a gram-positive cocci bacteria. If it is gram-negative, it is either Neisseria gonorrhea or meningitis. What you are looking at on this slide is a picture of a gram-positive diplococci arrangement of a strep throat swab. Different slides will have different colors and arrangements, but they will always have the circular cocci shape if it is actually a cocci. This slide is showing a picture of Streptobacillus mononiliformis from a patient's epidural abscess. It is apparent that it is Streptobacillus mononiliformis because of the way that the multiple rods are arranged in rows, making clusters. This is a gram-negative result, which is indicated by the pink color. It is more commonly shown in patients who have rat bite fever. This is the last morphology, spirilla. Remember, spirilla is only arranged in single arrangements. The picture you are looking at is a microscopic view of a Lyme disease slide. A spirillum bacteria called Borrelia burgdorferi is one of the infectious agents of Lyme disease. One of the most significant things about spirillum morphology is that they have a polar flagella, meaning that it can rotate the cell body in the opposite direction. Conclusion. From this lesson, I hope you now understand the history of cells, cell morphology, and the morphology and arrangement of cells. In a microbiology class, these topics are essentially the cells of understanding microbiology. If you don't learn about the basics, you can't further develop your understanding. Sometimes listening to lectures like this seem like they don't apply to your everyday life, but this stuff does. Every day, you are making and losing cells in your body while also coming into contact with thousands of bacteria that are fighting your own cells and trying to infect them. It is important to be aware of this and make sure you take care of your immune system so your fighter cells can be able to fight diseases. I hope that you enjoyed this lesson and learned as much as I did this semester about cells and microbiology.